All right. Hello, everyone. My name is John Hughes. I am the Director of Food and Agriculture Programs at UC Davis Continuing and Professional Education. I am also the Microbiology and Laboratory Instructor in our Master Brewers Certificate Program. And I'm delighted to be here today to share with you a presentation I've recently put together on yeast, a technical history. So, couple of things before we get started. One is I managed to catch some kind of nasty flu or cold at the Craft Brewers Conference last week, so I'm still slightly recovering. I apologize if I'm coughing or you can hear the cough drop that I'm sucking on throughout this presentation, so please bear with me on that. If you are participating live, I am keeping an eye on the chat, so feel free to put any questions or comments in there, and I'm happy to answer those as they come up. All right, so let's jump into it. So we've got a little overview. I always have to start with the history of microbiology, and brewing actually played a pretty significant role in that. So even beyond the brewing world, the germ theory and everything like that, that all owes its kind of foundations to brewing science. Then we'll get a little bit into the yeast basics, and then we'll kind of jump into the meat of things with the taxonomy and phylogenetics. If you're not familiar with that word, that's evolutionary history of yeast. And so we'll talk about the genus Saccharomyces. We'll talk about what the origins of lager yeast are, and then we'll get into a little bit on what is has been the domestication of yeast. So some historical figures and key events in history. First of all, always have to point out Reinheitsgebot. People always talk about this. The original decree in 1516 had 3 ingredients allowed in beer, which was water, malt, and hops. Yeast was not included in that. It was formally added in 1906 once the sort of full biochemical nature of yeast was understood. But the initial decree did not have yeast in it. Now, that doesn't necessarily mean that people didn't know about yeast. They didn't necessarily know what it was as a microorganism, but they certainly were aware that this sediment that formed in the bottom of a fermenting batch of beer was really good at leading to vigorous fermentations if they added that to the next batch. And to kind of illustrate that, the Old English word for this sediment was called God is good. And that is literally exactly what it sounds like. God is good. It was sort of a thought that God was leading to this fermentation, and that if you transferred this sedimentation into the next batch, then you were doing God's work, essentially. And so they had methods long before people understood what single-celled organisms were to harvest yeast and transfer them onto subsequent fermentations. So you can't talk about the history of microbiology without talking about this man right here, Antony van Leeuwenhoek. He was actually initially a draper. He dealt in tapestries, and he took an interest in microscopy because he wanted to look at the fiber count of some of the textiles that he was trading. And he didn't like the microscopes that were available at the time. And so he actually created his own lenses and created his own microscope, which he then later on was able to use to look at sort of aqueous samples. And he actually went down this huge rabbit hole of viewing all kinds of microorganisms. And so Antony van Leeuwenhoek is perhaps one of the first people to even really consider microorganisms and what their nature was. And he referred to them as animalcules, kind of like animal molecules. So this is a little diagram and an actual photograph of a microscope that Antony van Leeuwenhoek designed. It's a single microscope, so it's just a single spherical lens, very, very small. You can just sort of barely see it there. And then he had a little needle that you could just put a drop of water on, or he even looked at blood and various other bodily fluids. And then you would sort of hold this rectangular part up to your eye, and you'd hold it very close to the lens, and you'd be able to, it would greatly magnify what was in that drop of water. And so these animalcules, he was able to see blood cells. He was able to see yeast cells. He was even able to see some sperm cells. This is not the greatest image because he probably didn't really see many bacteria cells because, as we'll see, they're much smaller than some of these other cells. But basically, this started the interest in microorganisms and, wondering what these were. He published it, and people started looking further into it. So that was in the 17th and 18th centuries. Moving forward in time a little bit, Louis Pasteur, I'm sure you've heard of his name before. Perhaps most widely known for his role in developing the pasteurization process, which is to stabilize liquid and food products by heating them up. But he also was the first person to definitively demonstrate that yeast was responsible for fermentation. So he wasn't the first person to posit this. There were lots of people kind of pushing this around. People by the mid 19th century had seen these animalcules. They'd been seeing yeast, but there wasn't a strong consensus that the yeast was actually what was the cause of fermentation. And in fact, there were some very prominent scientists that were very rudely mocking those people who were suggesting the yeast were doing it. They published this kind of satirical paper where they talked about this yeast as a miniature factory that would urinate alcohol and defecate carbon dioxide or something like that. They just thought this was such an absurd premise. But Louis Pasteur, in his was the first person to really methodically demonstrate that it was yeast that was leading to the fermentation. And so he published this in a piece he called Études sur la bière. So he was French. That's studies on beer. And in this, he wrote, alcoholic fermentation is an act correlated with the life and organization of the yeast cells. And so how did he actually demonstrate that? Well, this little swan neck flask is how he did it. And so the way he would do this is he would take some wort and he would apply some heat to it. So this was sort of a round-bottomed glass flask with a really long neck. You could actually close this off or leave it open. And so in this, he would apply heat to it, warm it up, essentially pasteurize it, and then just let that sit, and there would be no fermentation happening. If he later took this and applied heat and then broke the top off, or in some cases, he would actually tip the flask over. And both of those situations that opened this up to the air, opened it up to dust that could settle in, which would bring with it microorganisms. And so these cases, there would be yeast and bacteria present, and they would lead to fermentation. And so by examining these, he could see that A, fermentation had happened, and that B, yeast cells were present. He could do it with, was able to do it with the presence and absence of bacteria. And so from this, he definitively showed that yeast led to fermentation. He also showed that bacteria were present in the spoiled beer, but were not present in the non-spoiled beer. So Pasteur had a very great effect on the kind of microbiology of brewing. Now, expanding on his work, we have this gentleman here, Emil Christian Hansen, who was an employee of Carlsberg Laboratory. That's of Carlsberg Brewing in Netherlands, I think. And so they actually had a very well-equipped laboratory. Emil Christian Hansen was the first person to actually isolate a pure culture of yeast. So he was able to separate out individual yeast cells by dilution, and then he was able to grow those up into full cultures. And so he was the one that was able to determine that there were some, quote, wild yeasts. So yeasts that were not our typical brewing yeast, and that they could lead to spoilage as well as the bacteria. So lots of people working on this stuff, really interested, and really built the foundation of microbiology as we know it, not just brewing. All right, so let's talk a little bit about yeast. Let's get into the basics, right? Yeast is a unicellular fungus, okay? So the kingdom fungi. And we've got ale yeast are Saccharomyces cerevisiae, and lager yeast are Saccharomyces pastorianus. So as my students know, I'm a total etymology nerd. I think it's actually really useful to be able to know what the word origins are, because then you can know what certain words are just based on knowing what the roots mean. And so Saccharomyces means sugar fungus in Greek. So saccaro, you've probably seen that, like saccharification in a mash. That's the generation of sugars. So saccharose is the sugar. Myces is the Greek for fungus. And then if we look at the specific epithet, cerevisiae is Latin for from beer. So think the word cerveza in Spanish, same root there. And then pastorianus is Latin. It's sort of in honor of Louis Pasteur. Saccharomyces cerevisiae is perhaps one of the most well-studied organisms in the world. And it is probably only surpassed by E. coli. Now, I have an asterisk there. Why do I have that? Well, it's not studied as a beer production microbe nearly as much as it is used as essentially a stand-in for humans. So it turns out that as the tree of life goes, humans and yeast are very closely related. Now, you might think that's pretty absurd, considering we are macroscopic, we have, sentience and all of that, and yeast is a single-celled organism. But when you get down to the molecular level, yeast and humans are actually quite similar, at least compared to a lot of the other organisms. So we know a lot about it. What has been kind of elucidated and studying yeast as a surrogate for humans has also helped with the brewing side of things. Certainly the yeast genome was sequenced much before we started even thinking about using sequencing in brewing. And that really led to a lot of insights in studying it as a brewing organism. Saccharomyces reproduces by budding. So you can see on this image here, these little tiny blebs that are kind of coming off. So this right here is a bud scar. That's where a cell is already budded off. And here is an actively growing bud. So whereas a lot of single-celled organisms grow by what's called binary fission, where they just sort of grow and grow and grow until they get to almost double size, and then they split into two equally sized cells. Saccharomyces instead buds off a smaller, what we call daughter cell. And then that daughter cell can grow into a larger cell and then can bud off daughter cells of its own. Saccharomyces can grow aerobically, that is with oxygen, or anaerobically. So that's what we think of as fermentation. And when it undergoes fermentation, it ferments sugars into alcohol and carbon dioxide. And so this is the chemical formula. Here is glucose, C6H12O6, and that forms 2 molecules of ethanol, or ethyl alcohol, and 2 molecules of carbon dioxide. All right, so here's a good graphical representation of what I was just talking about earlier. So one of my biggest pet peeves is when people refer to yeast as bacteria, and they are absolutely not. So this is the grand phylogenetic tree of life. This is all life as we know it on planet Earth. And so the way you can read this is the length of the branches is proportional to evolutionary history. So the longer the branch, the more evolutionarily distant those organisms are. So down here is the root. This is the single common ancestor of all life on Earth. There was a split event very early on, billions with a B years ago. And that created the bacteria. And then the other clade here is made up of the eukarya. That's us, fungi, plants, things like that. And then the archaea, which are some microbes, really interesting. Do not have time to get into that in this talk. But so what you can see is that the bacteria here are as far away, evolutionarily speaking, as you can get from the fungi, this green star, which is where yeast are. And then if you look, here are animals and fungi. We are in animals. And so fungi are our most closely related kingdom. So that's a nice little way to kind of show you how similar we are. So if you ever refer to yeast as bacteria in my presence, I will never speak to you again. No, I'm just kidding. But I do like to educate people on what the difference is here. All right, so we can split up brewing yeast into two different species. We've got lager yeast, that's Saccharomyces pastorianus. Sometimes this is referred to as bottom fermenting, although that's somewhat of an archaic term. And then we have ale yeast, which is the species Saccharomyces cerevisiae. So that one is called top fermenting. You tend to see more diversity in ale yeast compared to lager yeast. And that's really because they split, as we'll see later on in this talk, much earlier than lager yeast. And so they've had more time to undergo selection by humans than lager yeast have. Lager yeast consists of two primary lineages, which we'll see in just a second. And then a couple of differences that these are often used to sort of distinguish between ale and lager yeasts. If you don't know what an isolate is, for example, there's this disaccharide. It's a sugar made up of glucose and galactose connected together in a particular way called melibiose that lager yeasts are able to cleave and ale yeasts are not. So you can do a really quick test to kind of check to see what is what. The other thing is you can grow them at 37 degrees Celsius. That's about 99 degrees Fahrenheit. And ale yeast can grow at that temperature. Lager yeast can't. So really quick ways of distinguishing between the two. So of the different kinds of varieties that we have, lager yeast, there's sort of what we call the group 1 or group 2. Group 1 is also referred to as the Saaz type Bohemian lagers. And group 2 is the Froberg type or Bavarian lagers. So, for example, one strain that's in very widespread use today is the Weihenstephan 3470. That is a group 2 Froberg type. And in fact, most commercial lager strains used today are actually of this group 2 Froberg type. And they are, they ferment a little more rapidly. They are maltotriose positive. That is, they can transport and cleave maltotriose, the trisaccharide that comes from mashing. Whereas group 1 are not able to ferment maltotriose, and they do not ferment as rapidly. So just sort of the selection of being able to churn through batches rapidly and turn over tanks, people have kind of tended more towards this group 2 Froberg type. Flavor-wise, you might get a little more estery quality from the Froberg type, and that's probably due to their more vigorous fermentation, rapid growth. But not a whole lot of difference, certainly not the differences that you see in the different types of ale yeast. So, for example, when we think of ale yeast, we can think of the English strains, American strains, Belgian strains, the German ales like Altbier and Kulsch strains, Hefeweizen, and then the so-called wild Saccharomyces type yeasts, as well as wine, sake, and baking yeast. So if anyone has ever tried to make beer with a wine yeast or a sake yeast, you know that it doesn't always work out as well as you might like. You always get weird flavors. But there's a lot more diversity in the ale lineage than there is in the lager lineage. So let's talk a little bit about the taxonomy, and that's the classification of different species. And so this is, it's really hard to see this. We'll get a little more into this later. This is a phylogenetic tree of all kind of brewing important yeast, or really fermentation important yeasts. If we really zoom in on the Saccharomyces, or sorry, rather, this is a zoom out, but taking away a lot of the detail. On the Saccharomyces genus, we see that there are 4, 6, 8 species of Saccharomyces. So here's cerevisiae at the top. We've got some others like Paradoxus, Arboricola, eubayanus. And then we see these two over here. What's going on with these? These are hybrids. So we're going to talk a little bit about what hybrid yeast are. They're distinct species, but they're actually created by a mating event between two separate species. So in the same way, you can think of this as like a mule, where a donkey and a horse mate to create a new kind of animal that is distinct from those two parent species. Now, this is from a paper in FEMS Yeast Research. I always like to point this out. This is the breakdown of the Saccharomyces genus over the last 25 plus years. And you see there's, probably 15, 16 species in 1998. That's down to 7 species in 2003. We get some increase and then a slight decrease again in 2019. In all possibility, it's probably even changed somewhat since then. It's now been 6 years. This is not because species are being created or going away per se. It's not like we're having things go extinct. What's happening is that as science has gotten more sophisticated in sequencing full genomes and being able to compare organisms against each other, we've been able to kind of combine certain species into, multiple species into a single, or break one species out into multiples. And so things change as we learn more. And that's something that, the scientific process, right, is always changing. We are learning new information and applying that to what we have already known, and things are changing. So in all possibility, this will most likely change even going further, but we do know certain things pretty well. Saccharomyces cerevisiae is a pretty well-established species, and we know pretty well that Saccharomyces pastorianus is a hybrid. And if you connect this, it's a little difficult to see, so I drew some extra lines on here for you. It's a hybrid between Saccharomyces cerevisiae and Saccharomyces eubayanus. So, down here you'll see there are some synonyms here. So people at one point called this Saccharomyces carlsbergensis. This is not commonly used anymore. Pastorianus is the one that has been settled on. So how do these hybrids form? Well, remember I talked about how yeast reproduced by budding. When a mother cell makes a daughter cell, that is a clone. That daughter cell is a clone of the mother cell. It has exactly the same DNA, and it behaves in exactly the same way. Now, barring any mutations that might have happened, but there's no recombination that happens. But it turns out yeast are actually capable of sexual reproduction. And what happens here is that certain yeast will enter into a sexual stage of their life cycle. They will divide until they are they have half the number of chromosomes, and they will split into 2 different phenotypes. So we have the A type and the alpha type, and that's sort of analogous to male and female. So A type will only mate with alpha type, and so on. And that is just it has to do with some chemicals that they release that the A type is attracted to alpha type and vice versa. And so you can see here, this is a mating gamete that has this sort of appendage sticking out. So it's actually following a chemical gradient and extending that appendage out. And this is referred to within the scientific community as schmooing. So somebody saw this this old cartoon, and at the time this was given the name. It was not an old cartoon, but by Al Cap, and it was this weird creature that was kind of bowling pin shaped, and it was referred to as the shmoo. And so scientists have just as good of a sense of humor as the rest of us, and so they decided to call this a shmoo, and that made it into the scientific literature, and now people are calling that. So the two A type and the alpha type will shmoo, and they will meet together, and they will actually combine into a single cell. There will be some recombination between their DNA that creates genetic diversity, but it can also create a hybrid strain if the A type and the alpha type are from different species. So in this case, what happened was the one of the two types was Saccharomyces cerevisiae, and one of the two types was Saccharomyces eubayanus, and the resulting diploid daughter cell was Saccharomyces pastorianus. Now, I used the term diploid just now and haploid. What does that mean? That has to do with the number of chromosomes that a cell has. And when I say number of chromosomes, I'm really meaning number of copies of chromosomes. So yeast have 16 different chromosomes, just like humans have 23. But each of our cells has 2 copies. So we have 46 total chromosomes. Yeast cells have sometimes 2 copies, sometimes 3 copies, sometimes even 4 copies of their various chromosomes. So 16 different types, but then different copy numbers of each. So laboratory strains that people used to study tend to be haploid. They're easier to work with. You only have to make changes to one set of chromosomes. Baker's yeast can be haploid or diploid. Brewer's yeast are often what's called polyploid, meaning more than 2 copies, or actually aneuploid, and that just means you've got different numbers of copies of different chromosomes. We're not going to go too deep into that, but do know that there is a polyploid nature of most brewing yeast. So if we look at Saccharomyces pastorianus, we see our 2 types, group 1 and group 2. Group 1 is triploid. That means it's got 3N, or 3 copies of each chromosome. And it got 2 of those copies from the eubayanus parent. It's got 1 copy from the cerevisiae parent. Group 2, by contrast, is tetraploid. That's 4 copies of each, and it's got 2 copies from each of the parents. And so, you might be asking, how did we actually know this? How did we learn that this hybridization event even happened? Well, people had sequenced the genomes of these organisms, and just in looking at the total number of base pairs. So when you think of base pairs, remember DNA has the A, G, T, and C, and you count each of those individually up, and you get to Saccharomyces cerevisiae has 12.5 million base pairs. Remember base pairs because it's double-stranded. Eubayanus has 14.4 million, or mega base pairs, and pastorianus has 18.3. So different sizes here, but that doesn't tell us exactly what it means with their chromosomal identity and similarity. But when you looked at the percent identity, that is what DNA the species had in common exactly, Saccharomyces cerevisiae shared 58% identity with pastorianus. That means over half of their DNA was the same. Same thing if you looked at eubayanus and pastorianus, 70% of it was the same. So you might say, if if cerevisiae is like pastorianus and eubayanus is like pastorianus, then cerevisiae must be like eubayanus, but they only shared 10%. And so that indicates that you got some of the DNA from cerevisiae, you got some of the DNA from eubayanus, and they created their own hybrid progeny here. So where did this Saccharomyces eubayanus come from? This is a cryotolerant. That means it thrives in cold temperatures yeast. And it was first isolated on its own in Patagonia, South America, and Argentina in 2011. Since then, we've found isolates, and by we, I'm talking about the scientific community. I have not done any of this actual research. But we found some isolates in various parts of North America, Asia, and it actually has been found just as of a couple years ago in Europe. And this was pretty cool. This was a student project group at the University College Dublin. We share an acronym for our schools, but it was not UC Davis. And students were going around swabbing trees around campus, and they happened to swab this tree in particular. And when they streaked that onto a plate, they grew up Saccharomyces eubayanus. So whether this is something that got transferred over or where the original kind of actual parent cell came from is up for debate. For a long time, people were saying it came from Patagonia, and many of the Argentinians I know are holding that very closely to their hearts. They're saying we are the source. Some of the genetic and genomic data I've been seeing is potentially indicating that maybe the parent came from a Tibetan isolate, and so that was introduced to the European Saccharomyces cerevisiae via the Silk Road, as opposed to via the colonialists that were traveling to South America. So probably not something we'll ever really know definitively, although we might find some more information going forward. So this is from another paper in FEMS yeast research, and this is showing the possibility, the two different possibilities for how we came up with the two different groups of lager yeast. So remember, the group 1 was triploid; it had 3 copies of each chromosome, and group 2 is tetraploid, had 4 copies. So in one scenario, you had a hybridization of a diploid, 2 copies cerevisiae, and a diploid, 2 copies eubayanus. The hybrid was tetraploid, and then through some kind of action, you got chromosomal deletions, and one of them got a deletion in the chromosomes to get triploid, while the other one stayed tetraploid. So that's one possibility. Another possibility is that you had a couple of hybridization events, one between a haploid cerevisiae and a diploid eubayanus, and that created a triploid pastorianus lager yeast, and that was what was group 1. And then group 1 later had a second hybridization event with another haploid Saccharomyces to get to that tetraploid. Again, this is sort of best guess. We don't know. We probably, will never 100% know for sure. But these are two certainly very likely scenarios. Now that people know what the kind of hybridization was that led to lager yeast, some folks at Renaissance Biosci have created their own new hybrid. So they took Saccharomyces cerevisiae and Saccharomyces eubayanus and put them into mating states and allowed them to mate, and created their own novel lager hybrid, and they're referring to this as Nova Lager. They partnered with Lallemand to sell this. It's commercially available now. I've spoken with the folks at Lallemand actually, and they say that a lot of people are not using this to brew lagers. They're actually using it to brew things like IPAs. I think I heard brute IPAs, if anyone is still brewing those anymore, turn out really well with this new hybrid. Now, some differences. So this is now 3 sets of cerevisiae chromosomes and 1 set from eubayanus. So that's distinct from the group 1 and group 2. It actually has an increased temperature range, so it can grow warmer than other lager strains. And it was also created actually in a background strain that had a mutation that decreased hydrogen sulfide formation during fermentation. And that was actually unrelated to this project created at UC Davis by some yeast researchers on the wine side of things. So here is, to summarize, we've got group 1, group 2, and then what I'm now calling group 3, this novel lager strain. And you can see, you can compare the differences in where those chromosomes came from in all of these. So really interesting stuff. I don't know if there's anybody currently working on creating other interesting hybrids, but I'm sure we'll see something coming out in the future. All right, so moving on to the ale yeasts. I want to talk about this paper that came out just a few years ago, Domestication and Divergence of Saccharomyces cerevisiae beer yeasts. And so this actually took 157 different strains, analyzed them, sequenced their entire genomes, and split them up into different, what we call clades or clusters. And so they're grouped together by how closely they are related, evolutionarily speaking. And so they found 5 different kind of main groups, which they referred to as beer 1, beer 2, mixed, wine, and wild. So beer 1 comprises 64% of all brewing strains. So this is by far the workhorse. There are some brewing strains that exist in some of the others, including beer 2, but also mixed. But this is where you get most of them. And within this cluster, you see subclustering due to geographic separation. So you've got some Belgian and German clustering, you've got some British clustering, and some US clustering. And so this is, indicative of the kind of divergence of brewing traditions across Europe. And so these splits, the initial split to create this beer 1 clade probably happened sometime around 1600 AD. This was calculated based on the differences in the DNA in these yeasts by these researchers. So this is a best guess. It's not, there's not some definitive, this is the day this happened, but that's about when they think these diverged. And then the further diverging to create the beer 2 and wine yeast, 1650 AD. So some of the signs in this clade of domestication. So we can think of this in the same way we think of domestication of things like dogs or livestock. Certain traits will emerge that are beneficial to humans because humans are doing the selection here. And so a lot of this is the ability to ferment maltose sugar, right? That's the main sugar that is produced when brewing. One of the side effects is it doesn't handle stress very well. So think about if you took your dog and dropped it out in the Alaskan wilderness. Your dog was a direct descendant of wolves, and there are wolves in Alaska, and they survived just fine. Your dog probably would not do so great. No offense to your dog. My dog wouldn't do great either. But brewing yeasts are the same. They don't handle certain stressful situations better. They do handle the stress that we subject them to, which is high concentration of solvents like solutes like sugar in a wort, and ethanol concentration. So they handle those. We've selected the yeast to be able to handle those. But they don't handle really high temperatures, really low temperatures. They don't handle scarcity of nutrients very well. We've basically created this sort of pampered type of yeast that will do exactly what we want. If you contrast that with the wine yeast do not show the same signs of domestication. They can thrive in a wider pH range, in a wider ethanol concentration range. And the reason this is because if you think about it, wine is fermented once per year. And so people are not serially repitching wine yeast the way we are beer yeast. So there is no selection going on because there's no ability to kind of carry on yeast from one generation to the next. So you're going with the yeast that are naturally present on the grapes, or maybe you're pitching yeast, but there's no continued selection as we have in beer. And so if we zoom in on some of these included in beer 1 are the English ale strains, American ale strains, Belgian ale strains, the German ale strains. Hefeweizen is an interesting one. It's a mosaic. So it's kind of perhaps a hybridization between Saccharomyces, cerevisiae, and maybe something from one of these other clades. Could potentially even be a separate genus. So Terulospora could have actually exchanged some genetic information with Hefeweizen to generate this strain. Beer 2 is makes up about 20% of all brewing strains. There is no subdivision in here based on geographic limitation. So you find yeast that have originated in the UK, in the US, in Belgium, Germany, and Eastern Europe, but they don't cluster based on that geography. So this is suggesting that this split maybe happened later, and that this is probably having a lot to do with some of the kind of farmhouse strains that we're seeing. One of the things that we see in the beer 2 clade is that there are these genes called the MAL genes. These are for maltose and maltotriose utilization. And there is a transporter, the MAL11 gene, basically that encodes a protein that is able to transport maltose or maltotriose across the cell membrane. And this is defective in some of those strains, which makes them negative for maltotriose, or in some cases even maltose. The mixed clade here, which is more closely related to the beer 1 clade than the beer 2, is actually mostly bread yeast. So what we put into our dough to make it rise, 8% of brewing yeast out of this 157 strains that were tested exists within this clade. So probably got transferred over somehow. Maybe there was a brewery next to a bakery or something like that. But yeah, I wouldn't recommend using Fleischmann's yeast to ferment your beer, is what I'm saying. The wine yeast, again, I said, little sign of domestication. It looks like I got ahead of myself, and I already asked that question and then answered it for you, so we won't stay on this too much longer. The wild yeast show no signs of domestication, and these are just sort of environmental isolates. Their survivability in nature is high. One thing I did not talk about is sexual reproduction. So actually, many of the brewing strains that we have do not enter that mating phase of their life cycle. They have lost the ability to do so. And that is probably because brewers have selected yeast that don't change. They want the strains that make the same beer every single time they brew. And so that ability to kind of gain new genetic information by mating has been lost in a lot of these domesticated strains. You see more sexual reproduction in the wine yeast and in these wild yeast that show much less domestication. All right, so some folks decided that study did not go deep enough, did not look into some of the recent, or at least fairly recently popularized farmhouse strains like Kveik out of Norway. And it was actually found that those all clustered together, and that they, I think, ended up being most closely related, though still distinct from the Beer 2 cluster. The same researchers then also looked at some of the Baltic and Russian farmhouse yeasts, and those, again, formed their own clade, so there was probably some sharing done between farmhouses, and you got some changes just through genetic drift as certain farmhouses continued to use the same strain. Lastly, I want to point out there is a researcher named Christopher Krogerus who has a blog online called Shergork. And if you have a keen eye, you'll realize that is Krogerus backwards. And this guy, I would love to sit down and have a beer with this guy. He is a yeast, essentially a yeast genomicist, a yeast scientist who he's taken all of the publicly available yeast genomes and actually tied them with commercially available yeast. And so just like that grand brewing yeast phylogenetic tree that I showed you before, he's taken this and actually renamed them as what the commercially available yeasts are. So if we kind of zoom in a little bit, I hope this is legible. You can see we've got some WLP, WY, that's a Y yeast. WLP is White Labs. There is WB06, that's a Fermentus strain. This is a really interesting story right here. This WB06, for those of you who have brewed with this, you may be familiar. That was initially, is labeled and was released as a wheat beer strain, hence WB, intended to be used in things like a Belgian Wit or a Hefeweizen. And many people in the brewing community were saying, this is not making something that is really close to a Hefeweizen. This is actually very similar to the yeast used to make Duvel. So kind of a Belgian strong yeast. And as it turns out, actually, its most close related strain is this White Labs 570, which is the Belgian strong strain. And then if you look down next to the line, Y Yeast 1388, these are all Belgian strong strain ales. So I actually spoke with some of the Fermentus folks just at the Craft Brewers Conference and asked about this. And they said that it was historically, the Weihenstephan Hefeweizen yeast was not, at least the one that they had in their bank, was not particularly stable, and they made a decision, decades ago to release something different as their wheat beer strain, because they didn't trust it to kind of maintain stability on the market. They have since released, and I am sorry, I'm forgetting exactly what it is, but there is now essentially what is the Weihenstephan Hefeweizen yeast from Fermentis. Somebody knows that, maybe WB68 or something. It's kind of a fun story. It's the they got the permission to put the trademark on that from Weihenstephan themselves. So that's kind of a fun thing. You can find this whole tree at suregork.com, and you can explore this. If you ever have trouble finding a particular yeast, you can come on here and look at its closest related neighbor and maybe go with that instead. All right, so we're now in the present and future. So one of the big things that I see people talking about and using are the genetically engineered yeast. So these are companies like Omega Yeast and Berkeley Yeast. They're creating strains of yeast that are diacetyl free, or at least very low diacetyl. There's the so-called thiolized. This is yeast that are able to cleave thiols off of amino acids and make very potent tropical and fruity flavors. There's some yeast that have been engineered to create lactic acid. And then we're also seeing some low and non-alcoholic yeast strains being produced. So this is actually taking known yeast strains and knocking out, removing their ability to ferment maltose. And then I have my 4 question marks here. The sky is the limit. I mean, I know for a fact that both of these labs are working on new products. They won't tell me what it is. They won't tell the public what it is until they're ready to market. And, rightfully so. But I'm sure there's all kinds of fun and interesting stuff on the horizon. If you're not opposed to using genetically engineered yeast, there is nothing inherently dangerous about that, but I know some people feel kind of squeamish. But there's also, if you don't want to go the if you don't want to go the genetically engineered route, there are some options for lots of these different things. There are certain lager yeast, I believe, that are actually really produce very high amounts of thiol. And they have a very active beta lyase enzyme. That's the enzyme that cleaves the thiol off of amino acids. There are also a lot of different methods or strategies for making non-alcoholic beer. So there's maltose negative yeast naturally. There's also what's called Crabtree negative yeast. So this is kind of a fun one. If you've ever heard of the Crabtree effect, what this is if the sugar content is high enough, then in the presence of oxygen, yeast will still ferment rather than aerobically respire, even though aerobic respiration will produce significantly higher levels of energy. And it's thought that evolutionarily that's because the yeast wanted to make an environment that was difficult for their competitors to survive in, basically creating alcohol to drive off competitors. So that's the Crabtree effect. But there are Crabtree negative yeasts that don't have that effect. And so there are brewers that are bubbling through very small amounts of oxygen. I mean, we're talking parts per billion levels, right? And that's enough to prevent the actual fermentation from happening. So you're still getting all the metabolism other metabolism that's happening. You're still getting all those flavors that come from the yeast that make it taste like beer. You're just not getting alcohol produced. So lots of things happening. I think that this Lona from Lallemand is I want to say this is a new hybrid of different Saccharomyces strains, but that's used for low and non-alcoholic beers. But lots of really fun and interesting stuff on the horizon. And I'm always kind of trying to keep my ear to the rail for it. Always happy if anyone wants to contact me and share any fun stuff that you're doing or you've heard about. Definitely please share that with me. So with that, thank you for sitting and listening to me. Like I said, this was a really fun talk to put together. I am a self-admitted microbiology nerd, and yeast are pretty cool. So love talking about them. This is the Master Brewers Certificate Program class of 2023. I just want to point out there are 4 World Beer Cup medalists. Well, 4 Beer Cup medals, 3 medalists in this photo. So we have Marianne Ietsugu, who's a brewer at West Coast Brewing in Shizuoka, Japan, got a gold medal. We have Mark Berkowitz, who's a brewer at Original Pattern in Oakland, got 2 medals. And then Chris Moran at Fieldwork Brewing in Berkeley also got 2 medals. So 3 brewers from the class of 2023 collectively got 4 World Beer Cup medals. I'm so proud of them. You guys are awesome. And with that, thank you all. If you have any questions, feel free to type them in the chat. I think Andrew can add my email address to the notes on this video if you're watching later. So if you want to add that, you can contact me directly. Always love chatting yeast. But thank you very much all for your time, and thanks for being part of Craft Beer Professionals. And if anyone is interested in learning more about UC Davis continuing professional education and our professional brewing programs, I am always happy to talk about that as well. So thank you all very much, and have a great time.